Okay, let's start. So this is the last session of this subject for this week. And we will spend some time to finish the slides. So last time we stopped here. So I was explaining the model, the underlying model used by SCVI, and that is, as I said, the most important part of this lecture, the model assumption. And because once you understand what underlying model they use, then you can just go to use their Python functions, because all numerical issues have been taken care of by their function. So the key thing for a user is to understand what each parameter means. And so let's briefly review what we the SCVI model. So SCVI model is a model for the single-cell RNA seq data as counts. So it directly model the counts. So we denote the observed counts as X and G. So we have, suppose we have a big N times big G matrix containing N samples or N cells and G genes. And for each gene and each cell, we use a zero inflated negative binomial distribution to model that particular count. And in order to in order to fit so many distributions at the same time, we use a latent vector. So for each cell, we have a latent vector, and the dimension is often much lower. It's very low compared to the original dimension G of the problem. So, and in the SCVI package, it is said to be 10. So for each cell, we use 10 numbers, 10 latent variables to model its behavior. So essentially, we reduce the dimension of the problem from G to 10. So that's for dimension reduction, latent variable modeling. And in this distribution, we also explicitly model the batch index. This is for getting rid of batch effects. And we also model the library size, because as batch index, batch effects, library size is also what we want to get rid of. It's unwanted variation. So, and the core part of the model is a Bayesian hierarchical model. So we assume that the latent vector, the latent variables, follow a normal distribution, and the library size follows log normal distribution. And then the first neural network, FW, is used to model rho n. So rho n can be seen as normalized data. And then from this normalized data, we get negative binomially distributed counts. And from the counts, we can use, this is optional, we can use an additional neural network FH to model the dropouts. This is optional because it depends on data set. Sometimes if you believe in the data set there is significant dropout, then you should model that. Otherwise, you can just use negative binomial instead of zero inflated negative binomial. So to make, I briefly say, then the rest is to, now we have a model, and now how to make inference for that model, how to estimate that that model parameters in that model. And in the context of Bayesian statistics, here we are talking about posterior inference, because we have so the goal of the end goal of Bayesian statistics is to estimate the posterior distributions of the parameters. So here from now, if we look at the whole problem from an autoencoder perspective, then so what is an autoencoder? So the input of our problem is the counts. So as you can see here, this is we have for the nth cell, we have 1, 2 until G genes, G features. And then in addition to that, we have the batch index of the nth cell. So we know from which cell does the cell come from. And then we encode, so this is the encoder part. We encode or we reduce the dimension of the data from G to 10. So for each cell, we obtain a latent vector of 10 dimension, of dimensionality 10. And then we obtain an estimate of the log library size, L. And what about the decoding part? The decoding part is that from from this latent vector, we want to generate or regenerate the input. So why do we want to regenerate the input? Because in the process of encoding dimension reduction, so ideally we have already, but by doing this, we have removed unwanted variation, we have removed noise, because, well, if this is like PCA, so in the original data, higher dimension, we have a lot of noise. In order to use just 10 numbers, 10 dimensions to represent the original data set, we have to remove a lot of noises in order to compress the dimensionality from G to 10. So ideally, if we have successfully done that, then when we try to recover the data from these 10 dimensions, the data should be cleaner than the input. So, so this is a very typical autoencoder structure. By encoding, we mean dimension reduction. By decoding, we mean data cleaning. So, or essentially, a decoder is a normalizer. It normalizes your data. So the SCVI. So in terms of SCVI, the encoding part is to compute the posterior. So why? If you look at the model, then so this is the model is a negative zero inflated negative binomial distribution, and in this model we have a prior for Zn, that is normal, standard normal, multivariate standard normal. And the encoding part is that we want to, so from the data, the posterior distribution is a conditional distribution, which means, so on the right-hand side of the bar, we have Xn, which is the data, and the batch index. So from the batch index and from the data, we want to obtain information about Zn and Ln. So this step, encoding step, corresponds to this arrow here. So from Xn and Sn, we want to know the distribution of Zn and Ln. So this is different from PCA, because in PCA, I give you a data point, I give you a sample, then PCA, I give a data point to PCA, and PCA gives me a 10-dimensional or 3-dimensional summary. PC scores. But here, this is different. So instead of giving me just 3 numbers or 10 numbers, it gives me a distribution. So what do we mean by that? Because the prior distribution represents our prior knowledge about the latent variables, the latent vector. And before seeing it, because the latent variables are hidden, unseen, unknown. So before seeing the data, we actually know nothing about the latent variables. And then after seeing data, we use data and information from the data to update our belief about Zn. So that's the meaning of computing posterior. So here the posterior is the posterior of Zn, and Zn is the reduced, is the lower dimensional representation of the original sample. And the same after we have updated our knowledge about Zn, now we can sample from Zn, and based on this sampled Zn, we regenerate X and G. So this is the decoding part, and the decoding part is also probabilistic. Probabilistic here simply means instead of getting a set of numbers, we get a multivariate distribution. So essentially, instead of numbers, we get random numbers, or we get the distribution of those numbers. So this gives us more information compared to just non-deterministic, non-probabilistic modeling. So just briefly explain the, because I included a lot of details in the last few pages, but I don't plan to go through all of them. I just want to briefly explain that why the benefits of doing probabilistic modeling. For example, this we have already touched on dimension reduction. By reducing the dimension from G to 10, we have reduced the dimension. And the other thing we just talked about is normalization, because if you the output from the decoder is a normalized version of the original data. And because of everything is probabilistic, we can even use this model SCBI as a data simulator. So suppose we have trained a SCBI model from single-cell RNA-seq data. And then what we can do is we can sample from the posterior distributions. So we can sample from these distributions, and we can get however many samples we want sampled from this distribution. Because now that we know the form of this distribution, we can sample from it to obtain new data and new latent vectors. And then based on these, and then if we use these new, so suppose I write Zn plus 1, capital N plus 1. So suppose I generate a new latent vector from this distribution, and then by decoding, I can get a new data, I can get a new sample. So this is very useful, because that means based on the training result, based on the modeling result, we can not only estimate the latent vectors for the training data, but we can also generate new latent vectors, which which helps us to generate new data. So so if you look at, of course, if you look at those, the base, the foundation for generating those new data is from the training data. So essentially, we learn the information from the training data, and then we generate new data to try to mimic to using those information so that our new data should look like the training data. So this is a very natural way of doing simulation, and it's very and all these dimension reduction, normalization, and generation of new data are done under the same model. So that is the returning to the beginning. We talked about the motivation for SCBI. That is the workflow of single-cell RNA-seq consists of many different steps, but by using SCBI, we can do everything using the same model. And that is the strength of probabilistic modeling, that is Bayesian modeling. But the problem is that the problem of Bayes model is that it is more difficult to compute. So I have included a slide explaining that, but essentially, so the most difficult part of in a Bayesian inference problem is the computation of the posterior. Of course, we said that if we have computed the posterior, then we can do all sorts of things. We can do dimension reduction, normalization, and also generating new data, simulate new data. But the difficult part, of course, is to actually calculate the posterior distribution. And in order to do that, SCBI uses a technique called variational inference, and that's where the letters VI come from in SCBI. So SCBI stands for single cell variational inference. So variational inference means instead of calculating the original, instead of calculating the exact posterior distribution, we use an approximation. And this approximation is called variational inference. So we use a simpler, simpler, so it's here written as QZNLN. So remember that the the posterior is a distribution of Zn and Ln. So in order to approximate that, we use a simpler distribution Q, which is also a distribution of Zn and Ln. And we assume that and it is simpler because in this distribution, the marginal distributions of Zn and Ln are independent. So the joint distribution of Zn and Ln is just a product of Zn and Ln. And we assume the distribution of Zn is a normal distribution, and we can do similar simplification for Ln. We assume Ln, the posterior distribution of Ln, follows a log normal. So we use simpler, easier to compute distribution to approximate the more complex, intractable, much more difficult to compute exact posterior distribution. So we're doing an approximation. And since we're doing an approximation, we want to minimize the approximation error. So we want to minimize the difference between the approximation Q with the exact true posterior distribution P, and that is measured by a criterion called Kullback-Leibler divergence, KL divergence. And these derivations, by doing some derivations, we can show that minimizing the approximation error measured by KL divergence is equivalent to minimizing this term called elbow. So it's essentially a lower bound of the KL divergence. So so these are very detailed information. So I wanted to go through this just because this is a Bayesian inference process, and it's called variational inference. And at the end, we are trying to minimize a term called elbow, because you will see. So if you want to fine tune SCVI, you will see those terms. You will see likelihood. You will see elbow. So you will see those terms. So in order to understand the meaning of those terms, you just have to just look at this page. So let's look at some visualization results. We will look at a data set in more detail in the practical session after the lecture. But first, let's look at an example from the paper, SCVI paper. And this data set, and here we are applying SCVI to 3 data sets with different sample sizes. And here, the original paper is comparing 2 methods. The one is similar, the other is SCVI. So, as I said, SCVI can reduces the dimensions, and it can also be used as a normalization tool. So here, what we are seeing, 3 columns represent 3 data sets, and in the first one we can see that so this is the t-SNE plots for from the 2 methods after normalization, and the second column, from the second column, so for the first column, we can't really say which method performs better because all classes are well separated, and there isn't much difference. So, but from the second column, we can see that the t-SNE visualization for SCVI is much better than the other one, because you can see that this class is distorted, and whereas in the SCVI result, this purple part is connected to the main body of the data cloud, and this is better for trajectory inference, because you can see a clear trajectory from, because here we're comparing 2 cell types, and with so these 2 cell types are at the 2 ends of the spectrum. So if you want to do trajectory analysis based on this graph, then it's very hard, because first, as I said, you have 2 disconnected parts, and then the shift between green to blue is not very obvious. But from the second graph, you can see that the shift is very clear. So you can almost draw a line from here all the way to here. And then in the third example is mainly illustrating batch effects correction. So here we are plotting, on the left, we are plotting samples according to batches, and then on the right, we are plotting them according to cell types. So again, SCVI achieves better batch effects removal. Of course, this is a very crude way of examining batch effects correction, because typically we say that what we do is we use UMAP or t-SNE to visualize the data, and we plot, we color the data according to batch indexes, and if the data from different batches are mixed together, then we say they achieve the batch effects correction is good. This is just a very crude visual way of examining that. So we will leave more details to the practical session, because because here we're not doing much with SCVI. We just use it as a visualization tool. We can also use it for differential expression, but I will probably skip this part and jump directly into the practical section. Okay, so a brief summary about the pros and cons, a brief discussion about the pros and cons of deep learning based method. We have seen, we have reviewed the history of very briefly of deep learning, and we have introduced in detail SCVI. So according to my experience and also my discussions with colleagues, deep learning methods, what are the pros and cons? The good thing is, of course, it's very, for some tasks, deep learning is very good. It's very difficult to beat deep learning, especially when you only care about prediction. So, for example, in the imaging problem, we don't really do feature selection. So the input is a picture represented by a very long vector of pixels, and we just throw that into a new deep learning model, and the deep learning model tells us, the algorithm tells us what the picture is. Is it a dog or a cat or a house? So in this case, we don't really care about what leads to the prediction results. So is it because I observe a certain curve, or is it because I observe a certain color? So, well, sometimes we care, but often we don't care. We just want to know a result. Is this a dog or a cat? Then in this case, and if the data is very unstructured, unstructured means something like the picture, because when you when you flatten the picture into a long vector, you actually lose the original two-dimensional structure of the picture. So it is very hard to. So the data is very unstructured, or the other meaning of unstructured data is it's very difficult to extract useful features from the data. Because if you think about it, if you use a very long vector to represent a picture, then you end up having a lot of pixels, and these pixels are your features, and these features don't really have real world meanings. It's not like a patient profile where you have gender, age, ethnicity. These are all very features with good meanings. But in the imaging problem, you have pixels. So you don't really know what these pixels represent, except that maybe for color or light, light or darkness. So when you don't have very clear features, instead of raw data, instead you only have raw data, and if you only care about prediction, you don't care that much about interpretation. And also when you have a lot of data, and also preferably annotated data. For example, in classification, in imaging classification, you need a lot of annotated data. Then in this case, you can use deep learning. So large sample size, primary aim is prediction, and also data is unstructured. You don't have clear features to extract from. You don't know how to summarize the pixels into features, into more useful features. Then if this is the case, then it's good to use deep learning. And but the problem with deep learning is that the immediate problem is that it's very difficult to train a good model. like in modern imaging problems, when you deal with millions, billions of pictures, it takes a lot of time to train. And because for deep learning, the tuning parameter selection, parameter tuning is still a problem. You still need to tune your parameters. You need to select a structure, how many layers to use, how many nodes to use, and what learning rate to use, what optimization algorithm to use, etc. What is the batch, mini batch sizes, how many epochs you want, how many iterations you want. All these factors affect the results of the model training. So you still you have a lot of things to decide. And even if you know what are the good parameters, just going through the training process takes a lot of time. So it's very time consuming. You need very good computers. So that is a drawback. And another drawback is, of course, generally speaking, is more difficult to interpret. You don't know what happened between the input and the output. So it's not like a PCA where every PC is a linear combination of the original features. So you can see what feature, what gene is contributing to what, to which PC component. So yeah, so of course there are other pros and cons, but mainly the cheap ones, the main ones are these. And so in terms of single-cell RNA-seq and other data from other modalities, for example, single-cell ATAC-seq, nowadays deep learning methods are becoming more and more popular, but a big problem is how to, it's still a bit hard for biologists to use them because training takes time. Training requires expertise, and interpretation of the results requires expertise. So if you have a collaborator who is a computer scientist, of course that's good. But if you are just a biologist, you don't know much about these the mathematics, the computer science behind deep learning, then it's not necessarily a good idea to use deep learning. You can get some primary results by just using default setting, for example, as we will see. But if you really want to make the maximal use of deep learning, then you really need that expertise in computation. So, many people are doing researches on deep learning for biology. So we will see progress, perhaps, especially at least for imaging problems. Now people can rely on, most people would rely on deep learning, but for other problems where you have non-deep learning alternatives, I would suggest you to still be cautious, to really consider, why do I really need to use deep learning, and what it brings us in addition to conventional linear methods. Okay, so that is all for the lecture. And next we will go through the practical. Okay, so let's have a look at the main file. So if you download, so first you go through. So the idea for this tutorial is to, okay, so just briefly talk about the preliminary materials. The way I organize this practical is to use R, because SCBR is written in Python, but nowadays, data analysts, statisticians, data scientists are sort of divided because some really prefer using Python, some really prefer using R. So in order to communicate between these two languages, we have some very useful tools. So if you want to call Python functions from R, you can use this package called reticulate. And if you want to call R functions in Python, so if you're a Python user, you want to use R, there are some other packages. So I'm mainly an R user, so I would, when I have a data set, when I do some initial preliminary experiments, I always use R. So that's why and also for this subject, we have been using R all along, so, all semester. So that's why I use R. And you can see that once you have configured your computer, configured your R, then if you are familiar with the syntax of Python, then it's really easy to use Python in R. Okay, so I have summarized these in the document. So let's have a look at, okay. So in this practical, I have, another thing is to, you might want to install, because the original PCA function in Mixomics is a bit slow if you have really have a large sample size and a lot of features. So we have recently improved that. So if you install this particular version of Mixomics, then it's much faster. So first we need these packages, UMap for visualization and reticulate. As I said, it's for calling Python functions from R, and Surat data. So we're not really using Surat here, but we're just using a data set stored in Surat. And Plotly is for interactive visualization, and Scran is for benchmarking. So we will see, we will use Scran to normalize our data and compare it to SCVI. Okay, so first we set up our Python. That's because I didn't load these libraries, packages. So let's set up Python. And these import functions are just for importing the Python libraries. Okay, so here we're using a very popular, a benchmark data set, PBMC3K, and this data set, if you're using it for first time, you need to install, and then after that, it will be stored in your Surat package. So here I'm just because I have downloaded, so I can use it directly. And here I'm using an additional package, SC Easy. Well, there are many ways of doing the same thing. So here I just use SC Easy, which is from the Surat website. So here I'm using the convert format function within SC Easy, because the original format for PBMC 3K is Surat. It's a Surat object. But in order to work with SCVI, we need to use ANData. So ANData is a Python equivalent of Surat object, basically. Yeah, let's have a look. So ANData is 2700. So we have the number of organizations is 2700. And that is also different from, that is the main difference between ANData and Surat, because in Surat, the rows are the variables and the columns are the observations. But here in ANData is is more is using different conventions. So the rows are observations and the columns are variables. So we have 700 cells and 1300 genes. Okay, and we also have some metadata in it. So now we have loaded the data and we have converted to N data format. So these are, so I comment this. So these a few lines are for training. So in order to train SCBI, what you do is, so here the syntax is a bit weird, but it's not in, it's just mimicking Python, because in Python you would write scbi.model.scbi.setup and data. Okay, so here the $sign is equivalent to the dot operation in Python. Okay, so the first step, so essentially by doing this step, we are setting up the and data object. So our data and data object is a data, is named a data. And by doing this, we are telling this the package that we are using this. We will be using this as the training data. And then model is this. So this is our SCBI model, and data is the training data. And we're using 10 latent variables. And I put L here because if you put number L in R, you are telling R that this number is an integer. You have to do that here because otherwise, because essentially we are using Python by doing these a few lines. Because if you don't add L, then Python doesn't know it's an integer. Okay, so you need to tell Python that this is an integer. So we're using 10 latent 10 latent variables. So we're reducing the dimension from this to 10. And hidden units, so we're using one layer, one hidden layer for both encoder and decoder. So we have several neural networks in the model. We're using several neural networks, and for each neural network, we're using just one layer, one hidden layer. And for that hidden layer, we use 1 to 8 hidden nodes. So all these settings are the default settings. So I just the reason why I explicitly wrote them was because I wanted to show you what the default setting was. So these are actually the default setting, and you can adjust them by changing these numbers. So and then after you okay. So here we're just telling the we're not doing training yet because we're just telling Python that this is the SCBI model we want to train with these parameters. And then to train the actual model, we use model $sign or dot in Python train. So here I'm So in training, you can use a technique called early stopping. Early stopping means because if you don't do early stopping, then by default, the training will take 400 epochs. That means if you couldn't remember, go back to the slides about the training, deep learning training part. Essentially, we were telling the computer that we want maximum 400 iterations. But here by using, if you specify early stopping as true, then the computer will find a way to stop the training earlier. So often just 200, 100, because the idea is more iterations will lead to overfitting. So we stop earlier at some point when the validation error is not reducing. So if we see that the validation error, out of sample error, is not really reducing, then we stop the process to prevent overfitting. Okay, so so all these take time. So here we're dealing with a relatively small data set, and it takes a few minutes. But so here, in order to save time, I just because after you have trained the data, train the model, you can use this command to save the model, the training result somewhere, and then the next time you can just call, you can just load the trained model for direct use. So then let's get, so these few lines are for getting the output, the model output. So by using get normalized expression, you get the normalized expression. So essentially that is decoded data, normalized data. And that can, since we're in R, the decode data cannot be directly used because it's a Python object. So we use Py to R to convert the Python object containing normalized expression data to an R object. So now if we look at this always takes a bit of time. So okay, it's finished, and now you can see that the format is the type of the object is data frame. It's an R object, and the dimension of that is the same as before because we're not changing the dimensions, we're just normalizing. Okay, and the second, what's happening here? Get lead unexpected keyword and return dist. So if I set that to here, why is return dist? Okay, so here we're getting the latent representations, and we convert it again to our object. So now if you look at the dimension of latent, you have the same number of rows for observations for cells, and but you only have 10 variables. So this can be seen as a lower dimensional representation of your original data. So the next is visualization, because we have here we only, in this practical, we only talk about dimension reduction and normalization. We don't really talk about the differential expression, for example, and also simulation. So in order to, because it's easier to visualize data normalization and to visualize dimension reduction. So here, so the next step is to, so after getting those output, now we're trying to visualize them. And these three lines are just for UMAP. We are setting the parameters in UMAP. So in particular, this line, I set the number of components for UMAP as 3, because usually people by default people uses just 2. So you get a 2-dimensional plot, but here I'm using 3 because in my project I prefer looking at a 3-dimensional plots. And part 1 is to visualize the normalized data. So these data have the same dimension as the original one. Now, in the first step, in order to do UMAP, because UMAP can be very, very difficult, very time consuming when the dimension is very large. So here we're using PCA to first we bring down the dimension to 50. Okay. You can use more, of course, here to save time. I just use 50. Okay. So I have reduced the data from the dimension from 1300 to it's not 1300, it's 10,000. It's more than 10,000. Yeah, it's 13,000. It's 13,000 features to 50, and then I use UMAP to further reduce 50 to 3. You can see that even with 50 dimensions, it already becomes a bit slow. So it's a very standard procedure. You first use PCA to bring down the dimension to 50, 100, 200, and then you can compute UMAP. Now, if I use these commands to get the results, so these are the 3 UMAP components, and I'm coloring the data points by cell types. Okay, so these are the original cell type annotations, and so here I'm not doing any normalization. Here I'm just calculating the UMAP of raw counts. Now you can see that very clearly we have 2 big chunks here and here. So remember that in UMAP, the distance cannot represent, the distance usually doesn't represent the real Euclidean distance between samples. So we need to be very careful. So the distance is not realistic, but at least it tells, so what's important in UMAP is the neighborhood structure. Okay, neighborhood structure. So because UMAP tries to keep, because in the process of doing dimension reduction, you have to lose something. You choose to preserve something, and you choose to throw away something. So in PCA, you want to preserve the variance. You want the data clouds after dimension reduction to accurately represent the variance of the original data. And here in UMAP, you want to preserve the neighborhood structure. So if 2 points, for example, so any 2 points, so if 2 points here are neighbors, then they are probably also neighbors in the original data set if you consider Euclidean distance. And here we can see some points. For example, this is naive CD4 T cell, and this is B cell. So for this particular cell, because you can see that the majority of the green, the CD4 T cells are here, but there are some here. That means compared to the main body of CD4 T cells, this particular cell is probably closer in behavior to B cell, because if you look at its nearest neighbors, the nearest neighbors of this CD4 T cell are all B cells. So occasionally you can see some examples like this. And if we zoom in, we can see that the separation of these classes are not really good because we are visualizing raw data. And on the other extreme, we have monocytes, and so these are monocytes. So monocytes are separated from these T cells, and here is the B cells. So roughly, we have the desired, the sensible. We have a sensible dimension reduction results, but some classes are not very well separated. Now, next, let's use SCVI, normalized data. So we do the same thing, except that this time we are trying to visualize the decoded or the SCVI normalized data. Now we use the same code to visualize the data. Can we get a slightly better visualization? Okay, we have just finished PCA. Now it's computing UMAP. So different subtypes, different types of T cells are not well separated. So here you can see, so still we have three chunks: B cell, monocytes, T cells. So one thing we can immediately see is that on this UMAP, B cell is perfectly separated from T cells. So this is already this. Well, this is a sort of you can say this is a sort of improvement, because these two data types, major data types, are supposed to be very different. So we have a perfect separation, which is good. Of course, we have a B cell here, so this one is a bit special. These B cells, and if you compare different subtypes of T cells, CD4, memory, CD4, naive, CD4, CD8, and natural killers. So compared to the previous plot, these different subtypes, different kinds of T cells are better separated on the UMAP, because if you return to the previous plots, the separation is not that good. It's not that good. Here the separation is improved a bit. And then again, for monocytes, you have different kinds of monocytes are clustered together. So lastly, because here we are using SCVI as a normalization tool, so a natural question to ask is, how about we use other kinds of normalization tool? So we can use, for example, SCRAN. SCRAN is one of the most popular tools for normalizing single-cell RNA-seq. And most normalization tools try to remove the effects of library size, and that is the guiding principle of SCRAN as well. So now we use, so in order to apply SCRAN, we have to use, first we need to calculate something called calculate some factors, and then we use those factors to scale counts, the raw counts. Okay, so now it's done, and we have got normalized data, PCA, UMAP, and plot. Can we get something a bit different? So we expect, of course, we expect the SCRAN normalized data to be a bit better than the raw data, but is it comparable to SCVI? Okay. So here, okay, so here the pattern is very different. Okay, so the raw data UMAP and the SCVI normalized UMAP are relatively similar, but here we have something very strange going on. We have platelets here, and okay, so we only have platelets. Yeah, so these platelets are these 4 cells are not really clustered together. So if we look at the, so if we look at the SCBI normalized data, then the platelets are here and here. Yeah. So these are some sort of outliers. So we only have 4 platelets. So these are almost like outliers. But in SCRAN normalized data, the outlying trend has been exaggerated. So it's further away from the main body of data clouds. Now, if we, to make the visualization easier so we don't show platelets. Okay, so now we have, 3 clusters. So you can see that as CBI, B cells, monocytes, T cells. So again, we have that 3 clusters, major clusters. And then if you look at the T cells, then you also see something very similar to, so the 4 classes are relatively well separated, and you can see the same trajectory. And the monocytes are also clearly separated. So if we look at SCBI, the monocytes, it's similar. Maybe the monocytes, maybe SCBI hasn't really separated the monocytes that well, but the difference is really minimal. Okay, the only strange thing is that somehow SCRAN has exaggerated the difference between platelets and the rest of the cells. Okay, so that is one way of using SCBI, using it as normalization. What about a latent vector? So the first thing we do is we take latent vectors. We take the first 3 latent vectors, and we just visualize the first 3 latent vectors. Now, there is no special meaning about the first 3. It's not like in PCA, the first 3 PCs represent most of the preserve most of the variants, but here we don't really have that. So the first it's the same, taking any 3. Okay, not really special. So if we just take the first 3, then latent vectors, then doesn't really make sense, right? It's not very good. If we, as I said, it's the same. So if we take, for example, 1 x5 x10, so if we take these 3, then we get something similar. So this tells us that if we just take any 3 of the latent vectors, then it doesn't give you a good visualization. So what about we do PCA? So if we use PCA, so here we use PCA to bring down the dimension from 10 to 3. Okay, so we use a better way to combine information within the latent vectors to 3 dimensions for visualization. Okay, so here you can see that the classes are starting to be separated, and you might think that this is a lot worse than UMAP, because in UMAP we can see really clear separation of classes, but that's the nature of PCA. PCA don't give you, except that the different classes are really, really different. Otherwise, PCA doesn't give you that visual effects of perfectly separated classes. That's because PCA aims at different things. PCA tries to preserve the variance. Okay, it's not really like UMAP. So here we can see that, but you can see the different classes. So for example, here we have T cells, natural killers, B cells, So B cells, and this is monocytes. Yeah. So if we look at T cell, yeah, T cells are relatively close, natural killers. Okay. And then if we look at monocytes, yeah, this monocyte is away from T cell. Monocytes, yeah, again, monocytes are with monocytes. Dendritic cells, so these are dendritic cells. And platelets, yeah, platelets are very special. They are situated, they are located first. Yeah, these gray points, they are very special. They are relatively separated from other classes, and the B cells here. Okay, so we can still roughly see that three major clusters. So different major types are well separated, and the subtypes are not that well separated, but we can still gain the same information as from UMAP. So this is the PCA of, this is the visualization based on latent vector. Then of course we can also use the we can also use UMAP, for example. If we just use UMAP, now because latent vector only has 10 dimensions, so we don't need to do a further PCA. The only reason we did PCA before was to reduce the computational cost for UMAP. And now the good thing, so this is the good thing about using latent vectors to do visualization, because the dimensionality is already very low, so we can directly compute the UMAP. So if we do that, UMAP of latent vector compared to PCA of latent vectors, then we get, okay, we get this. This is a bit similar to SCRAN, actually. So remember, we have seen similar thing, similar trend. Okay, so if we delete those outliers, then we can see, yeah, again, three major cell types. And yeah, so DC monocytes, well separated. T cells are not that great, maybe, but again, very, very similar to UMAP of SCRAN normalized or SCVR normalized data. Okay, so now compared to, so the comparison I wanted to make is to the comparison between, so this is the PCA of latent vectors. But suppose we do PCA directly to raw counts, what would happen? Then okay, this is the PCA of the raw counts. Now you can see that we have some class separation, but you can see the T cells are not really, they're really clustered together. And also T cells are, yeah. The worst thing about this direct approach is that the T cells are clustered together with B cells. So you don't really have the separation between major cell types. Okay, monocytes are relatively well separated, but B cells are clustered together with some T cells. Okay. The other thing is, yet another thing is to compare normalized counts. So if we use, so this is SCRAN normalized data and PCA. So this is not UMAP, but just PCA. Okay, so you can see, well, this is PCA of square normalized data. You can see 3 clusters, major cell types. Yeah, T cells are relatively well separated, and monocytes, platelets. Yeah, this is also quite good. This is almost similar to the UMAP of some of the UMAPs. Okay. So in conclusion, I would say there are 2 ways of visualizing results from SCBI. One is you view it as a normalization tool, and you visualize the normalized SCBI normalized data. And for this particular data set, it achieves similar effects as SCRAM. It's a standard tool, so we can't really say SCBI is better than SCRAM. No, at least not based on just one example. So here we're doing a very simple benchmark experiment just to see what SCBI can do. And our comparison is, of course, very naive because we only have 1 lecture, 1 practical session. But one thing is that so why would we want to use SCBI? As we have seen, it probably doesn't improve much compared to SCRAM, for example. I would say it's good because its strength really lies in the fact that it does everything. So once you have computed the algorithm, you can use it to do everything. Because SCRAM is not necessarily good for differential expression, for example. If you want to do differential expression, you probably need some other normalization methods, because this is just visualization, okay? And visualization are limited to 2 or 3 dimensions, because we can only see 2 or 3 dimensions. But in differential expression, for example, we can still use SCBI, but you might need to use some other normalization methods, such as SC transform in SURAD. And also we're talking about latent vectors. So remember that we don't just have because what we visualize are sampled from the posterior distribution of latent vectors, and we still the good thing is that we have more than that. We actually know the posterior distribution of latent vectors, so we can do some more sophisticated things with those distributions, like we can sample new data, more data from those latent vectors, so to use it as a data generator, a simulator, essentially. So, and other possibilities, because the whole idea, the SCBI package is still under development. New tools are being added in. So, but once you understand the most basic one, you understand the others, because you see really similar terms. If you read their peak VI for ATAC-seq, they also talk about, it's the same thing. You still talk about likelihood, posterior distribution, latent vectors, etc. Except that for peak for ATAC-seq, you don't use negative binomial, you use Bernoulli. Okay, because it's about the data are 0-1 valued. Okay, they are not really about counts. So that's another good thing about this SCBI approach. If you want to use the same framework but a different data type, you change the probabilistic model, underlying model. For example, you change from negative binomial to Bernoulli. Okay, and if you have continuous data, you probably change negative binomial to normal. Okay, so yeah. Okay, so I think that's it for this week, and I hope this lecture and practical are helpful. I want to write it as when I wrote these materials, I really want to make them as useful as a future reference, not only just for you guys, but also for me, myself, because it's also very useful to review these things for my own research. Okay, so the key message today is that SEVR is probabilistic modeling. It uses deep learning. So what's the relation between probabilistic modeling and deep learning? I would say you have a probabilistic model and you want to solve it. You have a Bayesian probabilistic model. And the way you solve the estimation problem, the inference problem, is by using deep learning. So the deep learning is just a technique for solving this problem. So what's more essential is the problem, is the model itself. And then you are just using deep learning as a tool to solve the inference problem. Okay. And then the good thing about probabilistic modeling is that with one model, you can do a lot of different things and you don't need to switch between packages or tools. So that's very convenient. But the drawbacks of deep learning approaches, of course, it's difficult to train. And I didn't really show you the training process. It takes a long time. So yeah, so that is the take-home message for today. And I hope you had good luck with the assignments and exams. That's it for today.